Hello everyone. Today in this video I'm going to teach you Hybridoma technology for the production of monoclonal antibodies. Monoclonal antibodies are very important for disease diagnosis as well as the cure of the disease. These days the novel coronavirus 2 is spreading all over the world and it causing COVID-19, a disease, a pandemic disease. There's no medicine for that. How about this technology, hybridoma technology for the monoclonal antibodies for the disease? In this technology, there are a lot of steps. I will explain one by one. After this video, you can understand poly and monoclonal antibodies and you can explain hybridoma technology for the monoclonal antibody production, especially the coronavirus, and you can tell the advantage and disadvantage about the hybridoma technology. The first, you need to know what's polyclonal antibody. Actually, this virus, suppose the coronavirus enter in your body, it is a antigen. The body recognizes as antigen, and the coronavirus, they have a surface molecule like a spike protein, envelope protein, membrane protein. Each protein work as a epitope antigenic property. So your body will make B cells for each epitope and the B cells produce a specific antibody for all these antigenic molecules. So that's why the polyclonal antibodies are heterogeneous mixture of antibodies, recognize different epitopes on the same antigen in case of coronavirus, derived from multiple clone of B cells. So this is a polyclonal antibodies, and what we are concentrating is a monoclonal antibodies. So here you can derive only one type of antibody which is specific to only one type of epitope. This is very good for diagnosis as well as treatment purpose, derived from clones of single activated B cells, recognize single specific epitope and antigen, for example spike protein or membrane protein or envelope protein, and identical antibodies with the same specificity. That's what is very important for disease diagnosis as well as cure of the disease. And in this technology has been discovered by three scientists, George J.F. Kohler and Cesar Milstein in 1975, together with and Niels Jerne, and they produced this technology for monoclonal antibody production, and they got a Nobel Prize in 1984 because this technology worked in the world. So how this technology is working? First, you need to immunization of an animal. So in this case, we need to immunize this animal. For example, we need antigen. For in our case, we are taking novel coronavirus 2 and we are injecting into the virus into the animal. This animal get the disease, and this animal produce activated B cells, and the B cells will mature in the spleen. So if you take the spleen from this animal and if you crush the spleen, you can get the B cells, and you can isolate activated B lymphocyte which producing antibodies, which produce antibody against this antigen which is coronavirus. So here this all B cells produce many type of antibodies, so we get polyclonal antibody against this virus. This is the first step, and we need to collect this all activated B cells, so which is producing different antibodies, but we need to isolate only one B cells. Okay, so here, so activated B cell is producing antibodies against this antigen. So now the second, the cell fusion. Activated B cells are fused with myeloma cells. So we need another cell which is a myeloma cell. This is mutated cancerous B cell. Very important. This is again a B cell, but it's cancer B cell which is mutated. At the same time, cancer cell have the property that is divide indefinitely, but B cells, it won't. It has a little lifespan. it has a little lifespan, but in case of cancer cell, it has a long lifespan, indefinite lifespan. At the same time, these myeloma cells have some kind of gene mutation, which is HGPRT gene is non-functional. It means this enzyme is not produced in these cells, but activated B cell, this enzyme is active. Okay, do not produce antibodies. This is a B cell again, it won't produce antibodies as well as hypocysteine, guanine, phosphoribosyl transferase. This enzyme is absent. This enzyme is very important for salvage synthesis for the nucleic acid production. At the same time, every cell have a two pathway, salvage synthesis pathway as well as de novo pathway for the nucleic acid synthesis. So the myeloma cells cannot synthesize nucleic acid this salvage pathway, but this can use de novo pathway for the nucleic acid synthesis. Remember, activated B cell, it have this both pathway. Okay, remember this one. Then cell fusion. Now in the cell fusion, so we have activated B cells and we have myeloma cells. Remember, activated B cell lifespan is very low and it produces antibody. Myeloma cells, the lifespan is indefinite, and it has a HGPRT minus and immunoglobulin minus. Okay, so when we mix these two cells with a chemical called polyethylene glycol, this chemical assists the binding of cell membrane. So now I give you just a few seconds to think about how many cells will be produced after PEG treatment. How many different type of cells? So just 5 minutes. Tell me. Have you okay? How many cells have you think about that? One, two, three cells, four, five. So let me tell you. Total 5 different type of cells is produced. The first one is unfused. Actually, some cells will not fuse, and fused B cell, some two different type of B cells can fuse together, unfused myeloma cells and fused myeloma cells, and another type of cell is hybridoma. One is B cells with myeloma cells. So this is the one we are concentrating. This is the one we want, the cells with indefinite growth and produce antibodies. Okay, so now we need to select these cells from this, out of these 5 cells. How we can select? There's a selection of hybridoma. Yeah, another step, which is the third step, we use hat medium. The hat medium contains hypoxanthine, aminopterin, thymidine, three chemical together. In the three chemical, aminopterin chemical, which block the de novo pathway, by inhibiting the enzyme called dihydrofolate reductase. Okay, so the de novo pathway will be inhibited. So this chemical will will stop the growth of myeloma cells. At the same time, hypoxanthine and thymidine, these two precursor will be cleaved by this enzyme, HGPRT enzyme, and produce salvage pathway. This is used by B cells, and this chemical will kill myeloma cells. At the same time, this chemical will support B cells to grow. Okay, now the hybridoma cells will have this enzyme. Therefore, they only hybridoma cell will grow in this medium. Remember, in this medium only hybridoma cells will grow. Myeloma cells will die because there is aminopterin, and B cells will die after some time because the B cells have a low lifespan. Okay, so this is it. The selection of hybridoma. Fused and unfused B cells will die because the lifespan is around 2 weeks to 1 month. So only the B cells will die, and after some time the unfused myeloma cells also will die because they don't have this enzyme, and the aminopterin will block the de novo pathway. Okay, now the selection of hybridoma. So now we got 4 different hybridoma with polyclonal. It will produce combine all different antibodies against the coronavirus. So this is a mixed polyclonal antibody, but we need to select each cell by selection method. And here in the cell have a two property. One is indefinitely it will grow, at the same time they will continuously produce antibody. And selection of hybridomas, and after that isolation of hybridoma by single specificity character. So we can have a multi-well culture plate, and we can isolate individual B cells, individual hybridoma B cells, and we can uh, propagate separately, and you will have the monoclonal and different monoclonal antibodies for the detection of diagnosis of the virus as well as treatment. We can use this antibody binding with some drugs, and that drugs can kill the coronavirus. So this is a summary. So immunization of mouse, and isolation of B cells, cell fusion, and selection of hybridomas using HAT medium, and isolation of hybridomas with a single specificity B hybridomas, and screening of the product separately, cloning and propagation. Finally, we produce large amount of and monoclonal antibodies, different monoclonal antibodies for the cure and diagnosis of the. And there are some benefit as well as disadvantage. The benefit is we can diagnose disease, specific disease. We can use as disease therapy. Okay, at the same time it is very expensive and time consuming, sometimes give some side effect. So there are some defect is there, but we need to have some research on that. And so in this video we have learned something. So finally we got a medicine for coronavirus. Thank you. Any suggestion or any question, you can mail me. Thank you.